Casein agar. Testing for the hydrolysis of casein. Casein agar, also known as a milk plate, is a very simple test to run. You begin by drawing a line down the center of the plate. You will be inoculating the plate with two different bacteria. You'll put one bacteria on one side and one on the other. You will use aseptic technique to inoculate the plate with the two different bacteria. Here, bacillus is being inoculated on one side. Be sure to sterilize your loop in between inoculations. Now, Staph aureus will be inoculated on the other half of the plate. There is no need to stab or puncture the agar during inoculation. It is a simple, straight-line plate inoculation. Using a straight line will make the results easier to read after incubation. Once you've completed inoculation of the plate, sterilize the loop and be sure to label each half of the plate with the bacteria with which it was inoculated. Once your plate has been labeled, place it into the incubator until the next lab period. Shown here is an organism that is positive for casein hydrolysis. Casein is the protein in milk that makes it white. Some bacteria are capable of producing an enzyme known as casease that will hydrolyze the casein and break it down as a carbon source. Here on the left is an organism negative for casease, and on the right is an organism positive for casease. Here is another plate with a positive organism on the left and a negative on the right. Here is Staph aureus and E. coli. Staph aureus is capable of many different hydrolytic reactions, including the production of casease to hydrolyze the protein casein in milk. Casein as a whole protein is much too large for bacteria to take in, so the enzyme casease is excreted. It is called an exoenzyme. This enzyme will diffuse through the agar and break down any casein or milk protein that it comes into contact with. This forms the halo that you see surrounding the colony of bacteria. The observance of a halo around the colony of bacteria on a milk plate is positive for casein hydrolysis and the production of the enzyme known as casease. Casein agar, or the milk plate, is an easy and simple way to determine hydrolytic activity and the production of casease in bacteria. It is often used for identification of gram-negative or gram-positive organisms.